Welcome back to the deep dive. Okay, so today we're jumping into something pretty groundbreaking in gene therapy. It's about safely and precisely inserting really big pieces of DNA, like whole genes, into human cells.
Right, and the star of the show is this new system called evoCAST. The headline finding: it can slot in these massive DNA payloads. We're talking up to 15,000 base pairs.
Which is huge, gene-sized.
Exactly, and it does this with pretty decent efficiency, 10 to 30% in tests. But the really critical part, it does it without causing those double-strand breaks, the DSBs, that are such a headache with standard CRISPR.
Okay, that sounds like a big deal. I mean, how much better is it?
Compared to the original wild-type system they started with, it's something like a 420-fold average improvement. Just massive.
Wow. So maybe step back a bit. Why do we need this kind of gene addition therapy anyway?
Well, think about diseases caused by a broken gene, loss-of-function diseases. Often the best fix is just to add a working copy of the gene back in. It's mutation-agnostic, treats many different underlying problems.
But that's been hard, right? The existing methods.
Super hard. Things like homology-directed repair, HDR, they're just not very efficient, especially in cells that aren't dividing, which is, most cells in your body.
And they rely on making those breaks you mentioned, the DSBs. Why is that so bad?
Because when the cell tries to fix that DSB, it often messes up. It can insert or delete random bits of DNA right at the cut site. Those are called indels. Really dangerous, potentially causing other problems.
Okay, so DSBs are bad, but I thought there were other systems like CASTs, CRISPR-associated transposases. Did they promise integration without breaks?
They did. That's the potential. RNA-guided integration, no DSBs needed. But, and this is the key problem they tackled, the activity in human cells was basically nothing, less than 0.1% efficiency. It just didn't work well enough.
Right, so you have this tool that should work theoretically, but it fails in the human cell environment. How did they fix that?
They turned to evolution in a test tube, essentially, using a technique called PACE, Phage-Assisted Continuous Evolution.
PACE, okay, what does that involve?
It's a way to rapidly evolve proteins. They basically link the success of the gene editor, specifically the transposition part, to the survival and replication of a virus, a bacteriophage inside E. coli.
So survival of the fittest phage means a better gene editor.
Precisely. They knew the bottleneck was likely in the transposase machinery itself, the part that actually cuts and pastes the DNA. So they focused the evolution there, specifically on a subunit called TNSB.
The motor part, right?
Yeah, he called it the motor, and they ran this evolution cycle over and over.
And what came out the other end? What did this evolved TNSB look like?
Well, the best variant, called P415 TNSB, picked up 10 different mutations that boosted its activity. Just this evolved TNSB, when put back into human cells, achieved an average of 12% integration efficiency.
12% just from evolving that one piece. That's already way better than 0.1%.
Oh, absolutely.
Yeah.
And importantly, they did this evolution smartly. They made sure the system wouldn't rely on a bacterial protein called CLPX. The original TNSB needed CLPX, but CLPX can be toxic to human cells.
Ah, so the evolved P415 TNSB works without this toxic helper protein.
Exactly. It became CLPX independent, which is, pretty crucial if you're thinking about therapy.
Okay, this is fascinating. The system, evoCAST, is built around being safer because it avoids those initial DSBs. But let's pause here for a second. Thinking about the final product in the cell, what does DSB-free actually mean for the purity of the edited DNA? If you're not making that initial break, how clean is the result?
That's really the million-dollar question, isn't it?
Mm-hmm.
Especially for therapies. So they tested the final fully assembled evoCAST system. That's the evolved P415 TNSB plus some other engineered bits. And the results were pretty stunning. The average integration efficiency jumped again up to 19% across 4 different spots in the human genome. That's that 540-fold improvement over the original CAST system we mentioned earlier.
Okay, impressive efficiency. But what about the purity, the off-target effects, the indels?
This is the key part. They used high-throughput sequencing to look really closely. At the sites where the DNA didn't integrate, which is still the vast majority of target sites, right? They found no detectable indel formation. 0.
Wow. No indels at all.
None detected. That's huge. It means the system seems to be incredibly clean. It avoids those dangerous mutations that plague nuclease-based editors. The insertion itself was also super precise, landing exactly where it should, about 49 base pairs downstream of the guide RNA target, with single base pair accuracy.
That level of precision and cleanliness, that must open up a lot of possibilities, therapeutically speaking.
Absolutely. They showed some examples relevant to potential therapies, like they got 13% efficiency inserted a CAR construct into T cells, which is used in cancer immunotherapy.
Right, CRT cells.
Yeah. And they also efficiently inserted healthy copies of genes linked to genetic diseases like FANCI or MECP2, getting around 12–15% efficiency there. And importantly, this works regardless of the specific patient mutation.
And did they try it in actual patient cells?
They did. They used primary human fibroblasts from patients with a severe skin blistering disease, RDEB. Even in these sort of more difficult primary cells, they hit 19% integration efficiency, which is really promising for diseases where you need to correct genes in sensitive tissues without causing more damage.
Okay, so let's pull this all together. What's the big innovation here, and what does it imply for the future?
The core innovation is really taking this CAST system, which had huge theoretical promise but dismal performance in human cells, and using continuous evolution, PACE, to make it genuinely robust and efficient. They broke through that activity barrier.
And the implications?
Well, the DSB-free mechanism, combined with this high product purity, it makes evoCAST potentially ideal for applications where you absolutely cannot tolerate genomic damage. Think stem cell therapies or editing therapeutic cell lines like those CAR T cells. Plus, it's easily retargeted just by changing the guide RNA, like standard CRISPR.
All right, so if we had to boil it down, what are the, say, 3 key takeaways from this deep dive?
Okay, 3 things. First, evoCAST enables efficient, large-scale gene insertion kilobases of DNA into human cells, hitting efficiencies like 10–30%, which is a massive, roughly 420-fold leap.
Got it. Big jump in efficiency for large insertions. Second,
Its fundamental mechanism avoids double-strand breaks. This leads to incredibly high product purity, crucially, with no detectable indels at sites that weren't successfully integrated. Much safer profile.
no DSBs, no indels, safety. And third,
This breakthrough wasn't just luck, it was engineered. Specifically, using Phage-Assisted Continuous Evolution, PACE, to optimize that critical TNSB transposase component was the key driver.
okay, efficiency, safety via no DSBs, and PACE enabling it. Makes sense. But no system is perfect, right? Was there any mention of limitations?
Yeah, there was one important point. They did still detect some off-target integration events, meaning the DNA got inserted in unintended places in the genome sometimes.
Oh, okay, so not perfect precision yet.
Not entirely. They suggest this might be because the evoCAST machinery hangs around in the cell for too long when delivered via plasmids, as they did in this study. So here's a thought for you to chew on. If that prolonged exposure is the issue, could switching to faster, more transient delivery methods, maybe using mRNA or pre-assembled ribonuclear protein complexes, could that potentially reduce those off-target integrations even further and maybe, just maybe, speed up the path to clinical use?
Transient delivery to minimize exposure time. Interesting thought. Definitely something to keep an eye on. Thanks for walking us through that.
My pleasure. It's really exciting work.
Welcome to the Everyday Journal Club. I'm Hope, a biologist with 15 years of research experience, now fortunate to be working in a Nobel Prize-winning lab. This channel has 3 main goals. 1, to help you stay effortlessly updated on the latest scientific breakthroughs. No paywalls, no jargon overload. 2, to build a supportive community where we can exchange the most valuable experience-based research advice. 3, to share valuable strategies and concrete examples on how to achieve meaningful scientific breakthroughs. Once we reach 1,000 subscribers, I'll begin releasing content focused on the 3rd goal. Let's go! Let's make great science together.